Secretary for today's program, it gives me immense warmth and pleasure to greet all of you here. On behalf of the management, staff, and students of KVSR Siddhartha College of Pharmaceutical Sciences, a genial welcome to one and all for the online faculty development program on emerging trends in pharmaceutical sciences, challenges from academic research to commercialization, organized by Department of Pharmaceutics and Biotechnology in association with Siddhartha Innovation and Incubation Center, KVSR Corps Vijayawada. Today, in this online platform, we have professors, faculty, research scholars, and PG student fraternity from various institutions all over the country. Firstly, I would like to thank each and every participant for showing their keen interest to attend this program for acquiring the knowledge in the recent advances in the field of pharmaceutical sciences and research. Let us start the program by supplicating the Almighty with a prayer. I request host to play the prayer song. Please play the song. Song play out. Song play out. సార్ వీడియో దగ్గర పెట్టి ప్లే చేయండి సార్. I express my heartfelt gratitude to all the eminent speakers over here who have accepted our invitation to enlighten us in the various aspects. Education plays an important role in enabling a person to face a real-life situation with adequate knowledge. KVSR Corps is committed to the education of pharmacists with highest ethical standards and professional standards. Now I request our young and dynamic principal, Madam, a woman with a great vision, profound knowledge, focused, and a strategic leader, and who has a passion for excellence in everything she does, to present the inaugural speech. Over to Principal, Madam.
Thank you, Arifa. Honorable Chief Patron, Dr. C. Nageshwara Garu, President, Siddhartha Academy of General and Technical Education, Patron, C.P. Lakshman Rao Garu, Secretary, Siddhartha Academy of General and Technical Education, Convener, Sri C.H. Arun Kumar Garu, KVSR Siddhartha College of Pharmaceutical Sciences, distinguished speakers, faculty members from various colleges, and other delegates. A very pleasant morning to one and all. This is Professor A. Sunita, Principal, Head Department of Pharmaceutical Analysis, KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada. I am pleased to take this opportunity to extend a hearty welcome to all the eminent speakers and faculty members on day one of Faculty Development Program. Siddhartha College of Pharmaceutical Sciences feel privileged to inaugurate 1 week Faculty Development Program on emerging trends in pharmaceutical sciences, discussing the challenges in commercialization of academic research from 18th July to 24th July 2022, which is organized by Department of Pharmaceutics and Biotechnology. The main objective kept in mind while framing this faculty development program is to answer some of the questions like why commercialization of research work is not being done, what is the role of research work commercialization in improving pharma sector, and what sort of emerging technologies need to be implemented in order to improvise the functioning and efficacy of pharma industry. Well, the future technology is running around robotics, artificial intelligence, machine learning, 3D printing, precision medicine, and patient design. These terms might look like jargons of computer science, but now let's think big. Why are we not implementing these technologies to improve the functioning of pharma industry? Reason behind it is lack of awareness, knowledge, and proper guidance. It's we, the Faculty of Pharmacy, need to be updated on the emerging trends in our industry and encourage the students to pursue their interests towards this. Only then the future pharma birds can make these technologies implemented and enhance productivity and management of pharmaceutical domain. I hope this 1 week faculty development program will definitely provide a beneficial knowledge on emerging trends in pharma and challenges in commercialization of research work. On day one of this FDP program, here we have our today's distinguished speakers, Dr. Lingaya Garu, Professor and Senior Principal Scientist, CSIR, Indian Institute of Chemical Technology, Hyderabad, and Dr. ZV Murli Mohan Babu Garu, Advisor, Formulation, DEXA Development Center, Jakarta, Indonesia. I hope this program will be definitely helpful to the all the pharma faculty. I thank all the eminent speakers for accepting the invitation on behalf of KVSR Siddhartha College of Pharmaceutical Sciences and putting up your interest to share your knowledge with all the faculty members. I thank the management of Siddhartha Academy of Journal and Technical Education for their immense support to organize such beneficiary programs to all the pharma fraternity. A special appreciation to the program coordinator, Dr. Ramana, Head Department of Pharmaceutics and Biotechnology, Program Convener, Dr. Sheikh Arifa Begum, Assistant Professor, Department of Pharmaceutics, all other organizing committee members, and Dr. Ravi Shankar for his technical support, and the faculty members of KVSR Siddhartha College of Pharmaceutical Sciences. Thank you one and all for giving me this wonderful opportunity.
Thank you for your kind words, ma'am. So, let us now have a brief video of all the eminent speakers for this FDP. Darshan sir, please play the video. కనిపిస్తుంది సార్ షేర్ చేయండి షేర్ చేయండి షేర్ చేయండి
No, it's not sharing. Yes, it's connected.
Yes, no, ma'am.
Yeah. Cordial welcome to Dr. Lingaya Garu, session
you,
one
ma'am.
speaker. Extend my hearty welcome to Mohan Babu Garu, Dr. Girija Petro Garu, Dr. K. Kannan Garu, Dr. Anubhav Khale, Dr. Falguni Pati, Dr. R.K. Sareen, Dr. Adi Narayan Reddy Garu, my special thanks to all the speakers who have accepted our invitation to share their knowledge. Thank you, Arifa, for sharing this tool.
Yeah.
One second.
Arifa, unmute yourself and invite Dr. Ramana.
Now I request our HOD sir, coordinator of this FDP program, encouraging and supportive to enable us work in a very conducive manner, very down to earth, an incredible human being, Dr. G. Ramana sir, to address the gathering. So please address the gathering.
Thank you, Arifa. So, dear participants, our young dynamic principal, Dr. Sunita Garu, and our faculty members, and my dear friend Lingaya Garu, very good morning. So, first of all, we are very thankful to our principal, Dr. A. Sunita, and Siddhartha Management for their continuous support for conducting this kind of programs. So, the Department of Pharmaceutics and Biotechnology, in association with SPIC of KVSR Crops, is organizing a one-week online nation-level faculty program on emerging trends in pharmaceutical sciences, challenges from academic research to commercialization. So the Kommareddy Venkata Sadasiva College of Siddhartha College of Pharmaceutical Sciences was established in 1994 and is affiliated to Krishna University, Andhra Pradesh. This college is a self-financed institution by Siddhartha Academy of General and Technical Education, which is a non-profitable organization. Our college offering B.Pharm course, M.Pharmacy branch, 2 or 3 branches, and also PharmD course with PB also. The Department of Pharmaceutics and Biotechnology had received a worth of 2 crores funding from various funding agencies like DST, CERB, and AICT. So already the main objective of this FDP is to encourage faculty towards academic research in contemporary times as well as constructive academic collaboration to the industry. So dear participants, so we have excellent speakers from this FDP from different places around the India. Dear participants, utilize this opportunity, interact with them and acquire knowledge. And one more important point, daily attendance is compulsory. And at the end of FDP, so you have to submit feedback form, then only you will get the participation certificate. So this FDP is planned for 7 days. Totally 8 sessions are there. The first session, the speaker is Dr. Lingaya Nagarappu Garu. Session 2 is Murli Mohan Babu Garu from DEXA Development Center, Indonesia. And third session is Girija Patra Garu from Mumbai. Dr. Kannan is the fourth session. And fifth session is Anubhav Khale. And session 6 is Falguni Pati, is from IIT Hyderabad. And seventh session is the R.K. Sareen, professor from National Forensic Science University, Goa. And eighth session is given by Dr. Adi Narayan Reddy Garu, Dr. Reddy Laboratories, Hyderabad. So, dear participants, please utilize this opportunity and acquire knowledge. And over to Arifa. Voice not loud.
Wi-Fi is just muted. Ask her to unmute. Yeah, now it's on.
Now I extend hearty welcome to Dr. Lingaya Nagarapu Garu for accepting our invitation present for this MPP. Now I invite Dr. G. Ramana sir to introduce today's session. First session eminent speaker Dr. Lingaya Nagarapu Garu. So please introduce Dr. Lingaya Nagarapu Garu.
Okay, welcome to 1 week online faculty development program. So the first session today you are going to start. So the first eminent speaker is Dr. Lingaya Nagarapu Garu. So he's going to deliver a topic on Indian pharma vision, innovations and their impacts. So it's my pleasure to introduce my friend and great eminent scientist, Dr. Lingaya Nagarapu. So presently is holding as a senior principal scientist position at the CSIR IICT, Minister of Science and Technology, Hyderabad. He is also professor for ACSIR, New Delhi. So he bagged many awards. He received Best Scientist Award from Science Tech Foundation, Bangalore for the year 2013. And also he received CSIR Technology Award in 1996 from Director General CSIR, New Delhi. His major research is focused on design and synthesis of bioactive molecules such as adjuvants, anti-inflammatory, antibacterial, anti-cancer, and anti-tuberculosis molecules. He also majorly involved in applied research for the process development for 15 technologies. His research contributions, so sir has guided 30 PhDs and 50 MSc theses and 55 MPharmcy degrees. He published more than 120 research papers in reputed national and international journals, and sir has the many patents also. So the total impact factor is 250.378, and the citations also more than 4,000. Sir has reviewed 110 PhD theses and conducted 96 PhD viva voce from various universities and institutions in India. So other contributions, so for upliftment of science and technology in country, sir has delivered more than 60 invited lectures as a chief guest and a keynote speaker in national and international conferences on Indian pharma vision, which were developed by his research group. As a visiting scientist, had been to Department of Pharmaceutical Chemistry, Yangon University, South Korea. And also, sir is a member, Pollution Control Board, State of Telangana. He is a chairman and Science Society, Hyderabad, and member, Royal Society, Chemistry, London. And is the other and drafting committee member of chemistry syllabus for Dr. B.R. Ambedkar Open University. And is a chairman, governing body of Mallareddy College of Pharmacy, Hyderabad. And also, he started having many key positions. He consultant to a couple of chemical industries in Hyderabad and member well-wisher committee for internal implementation and progress review of program, Osmania University, Hyderabad, board of studies and subject expert to Krishna University, say Krishna Devaraya University and JNT universities in Hyderabad, also Andhra Pradesh also. Subject expert for Department of Chemistry, Department of Pharmaceutical Chemistry, in Osmania University, JNT University, Hyderabad, and JNT University, Anantapur. He's a review for national and international organic and medicinal chemistry journals. So with this sound biodata, I welcome you, sir, to continue the session to speak on this topic.
Yeah. Thank you, Professor, for your nice introduction. And can you able to hear my voice clearly?
Yeah, hearing, sir.
It's okay.
It's audible.
Okay. Very nice, very nice. Just I'm sharing my slides. 1 minute. 1 minute.
Yes, sir, it's connected.
It's connected, ma'am?
Yeah, maximize
Yeah.
the screen. Please.
Okay, just it's opening. It's little bit taking time.
Yes.
No, it's okay, ma'am. It's perfectly.
Yeah, it's good.
My voice, everything is okay.
Clear.
Yeah. Now, let me thank the principal, the eminent personality in that chair, Dr. Sinthe Garu, and my dear friend, the Professor Justice Ramana, who has given this opportunity to say the few words about my journey particularly in the CSR, particularly where we people are working on the Indian Pharma vision fulfillment. But in that, I'm going to just convey, innovations and their impacts. If you're looking at the way you people are selected for the FDP programs, really emerging trends in pharmaceutical sciences, that's given the general. There, I impressed a lot. Challenges from academic to commercialization, academic research to commercialization. Honestly speaking, the word, but real, this is the first time hearing. My journey is more than almost 33 years in the research area. But this first time I am saying this one. The first time I'm saying this one. Where the call pharmacy, the pharma call is talking about challenges from academic research to commercialization. Even just our respected principal also is mentioned in his speech. Why we are unable to go for the commercialization? Just a minute. Just I want to take my just official call. Is there?
Okay, sir.
Tami. There is Sanjita Market, everything is okay. And okay, lecture because I'm checking the message, but it's okay, it's no problem. Okay, okay. Sorry for the interruption. Just there was a call from my CSR headquarters. Yeah.
Okay,
Regarding the title, ma'am. Regarding the title, I impressed. Challenges from academic research to commercialization. As per the commercialization concern, the national institutions such as IACT, again, IACT is the Pune, NCL Pune, we the leading persons in the commercialization of the products. Basically, academic research, yes. If you look at this, you take any molecule, the developed by the any eminent personalities, any industries, any pharma sector. Fundamentals come from the academic only, from academic research only. But you people are concentrating at the academic level to commercialization. If you're able to do such type of research at your pharmacy just college level, the country will appreciate you. The country will give you a lot of appreciation, because you people are the talking where you people are running a pharma institution. The pharma industries, the people are talking about this, commercialization, okay, that's quite suitable. It's a normal work for them. But at pharma institutions level, where you people are concentrating academic research to commercialization. Really, being a national scientist, really I'm personally appreciating this title, ma'am. That's what my guest is again. Really, I'm impressed. Then coming to the my talk. So, being a national scientist, up to 2023, every scientist from the national laboratory, we used to talk about the Indian pharma vision. The particular today, I also selected this one, innovations and their impacts. Again, what is Indian pharma vision? First, we should know. Then only we can see what are the innovations that in fact the lying at present in India. What type of research is required? What country is science and technology waiting for the young researchers? Whether it is the pharma sector, pharma institutions, and pharma industries, both ways, what it may be. The Indian pharma is waiting for, where Indian pharma vision is lacking 20% less. Now, let me present. Today, my simplify by outline of my work case, Indian pharma vision, their facts, then information about drugs developed by India so far, then present status of Indian cancer by World Health Organization, then importance of the open source drug discovery towards the anti-tuberculosis program in India, and the role of CSR IST in generics and healthcare sector. Being a national scientist from the CSR IST, but I need to tell what's our contribution to our national progress of the nation. That's it. Then some of my research findings towards the drug discovery program, where we people are talking about the Indian pharma vision. So what type of research is required for the fulfillment of the Indian pharma vision? In that aspect, being a CSR scientist, there was some my contributions. So I given the last way my research findings towards the drug discovery. Now, let us concentrate first innovations towards the Indian pharma vision. Before going to innovations in India, but we should know what is the Indian pharma vision. Particularly, we should know. Every Indian pharma citizen should know what is the Indian pharma vision. Then only we can fulfill our dreams. We can fulfill our contribution toward the research. Now look at these are the young stars dreaming about the fulfillment of the Indian pharma vision, because still the India is lacking 20%. We are behind of the 20% world market. As such, why 20%? We need to talk the 20%. If you look at the statement of the Indian pharma vision totally, what happened? The first, global production of the pharmaceuticals, the branded and generics put together, more than $550 billion. But the Indian pharma industry at present has a turnover 30 million of the generics and branded products. While evolving the pharma vision 2020, here up to we people, Indian scientists should talk about the 2023, because still we are behind of the 20% less. The pharma vision is supposed to be fulfilled by 2020 only, but they extended due to the less number of the generics and branded. We should identify all such missions which will make India the leader in drugs production. For that, what I have to do? We should set a target of the production of 20% of the world generics, where India has got a core competence producing the cost-effective drugs. Of course, no doubt, the Indian pharma vision looking for the 20% less. It should be total $50 billion of the market. Then only the fulfillment of the Indian Pharma Vision 2020. But we are behind of the 20% of the world generics and branded drugs. Okay. The Indian Pharma Vision waiting for that to 20%. But whatever we do the 20%, it should be cost-effective pharma products. Due to the Indian poverty, below poverty, I mean 80% of the Indian population, looking at the Indian population, where rickshaw pullers and daily wages unable to buy the costly drugs. The market is providing. The market is providing. Pharma market is providing. But due to the below poverty of the country, people are unable to buy it. This is the reason whoever do this going for the even as a basic research or the commercialization. It should be cost effective. Your drug, whatever you're going to develop the research, the fundamental research or the conventional drug, it should be cost effective. Then all that cost effective drug should be 20%. That 20% going to be added in the 30% market. The totally $50 million market. Then India would be a competence of producing the cost effective research, a great leader in the producing the world market. This is an Indian pharma vision. The science and Indian scientific technologies are waiting for that 20% should be cost effective. That's the step of the research is increasing in India at present. That sort of research is required. If you look at the small statement, after independence, India has developed 21 drugs. Why I'm talking means after from the independence to till today, but we should know what are the $30 million market is going on so far. This is 21 drugs. Still, 20% more is cost effective required. So far, after independence, only 21 drugs has developed. Okay, but here look at what are the 21 drugs. In 1960, the first drug, that's a hamacin, which will be used for the fungal infection in the mouth. In 1960, first drug was developed after independence. I'm talking here. Innovations, no? I've given my title, Innovations and Their Impacts. A couple of minutes back, we have seen what is Indian Pharma Vision 2020. In that, we are behind the 20%. That 20% if we are able to produce, that should be cost effective. That tells about the Indian Pharma Vision. Now, coming to the innovations. After independence, India has developed 21 drugs. The first drug developed in 1960, that is hamacin. Then 20th drug was developed in the 2014, that is lipaglyn, which will be used for the diabetic dyslipidemia in India. Okay, 1962 2014, 20 drugs. There was one specific drug, 21st drug, which was developed in the 2019, that is Kudos CM9. This is an anti-cancer drug. The one more forward moment to talk about 14 drugs out of 21 developed by my parent organization, that is CSIR, including the IICT. Where we are talking about the 21st drug, Kudus CM9, no, recently they developed the anti-cancer drug. That was also developed by my CSIR only. That is Indian Institute of Chemical Biology, Calcutta. So These are the major contributions towards the country development, particularly the CSIR. Now, let us see, what are those 20 drugs where the India has developed after independence? If you look at in 1960, there was a first drug, that is Hamacin, which will be used for the oral thrush, that developed by the Hindustan antibiotics. Then similarly 1962, 1988, there are few drugs. The latter followed by again 1989, 1992, 2014. I told you no. 20th drug was Lipaglyn, which was approved in the 2014, which was developed in the 14. So ZS Cadila Ahmedabad, the company was developed that 28 drugs. Later, if you see, in 2011, it was one efficient drug. That drug name is called Syndriam. That cure fast malaria was developed by Ranbaxy in the collaboration with the Department of Science and Technology. Then the Ranbaxy has introduced in the market to the adult patients. As and today, that very efficient drug is available in the market. Why I am specifically talking about the Syndriam drug? Because that Syndriam drug was developed by the Ranbaxy for the fulfillment of Indian Pharma Vision 2020. In that vision only, keeping in the view of the Indian Pharma Vision, the Ranbaxy has developed that Syndriam drug with collaboration Department of Science and Technology. This includes that 20 drugs. Now we are talking about the 20. I told you that I specifically mentioned about the 21st drug. This is developed by again my CSR family, Indian Institute of Chemical Biology, Calcutta. The specialty of the drug is, this is first world Ayurvedic anti-cancer drug. You might have heard about the anti-cancer drugs from the synthetic way, but this is the first Indian, this is the first world Ayurvedic anti-drug. Enter in the world. There was an Ayurvedic drug for the anti-cancer, but our India has developed that anti-cancer drug, but the Ayurvedic anti-cancer drug. Then what are that specialty? What that specification indicates? That means it's a non-chemical, non-synthetic Ayurvedic drug. The result of 30 years of extensive research by the Indian Institute of Chemical Biology. You know, for the drug development, it will take us more than 30 years. But recently we have taken the vaccines within the short period where India, including India, has developed. India and other countries also developed that vaccines, Covaxin, Covishield. But if you look at the particularly Kudus CM9, no side effects, proven to be the most effective, the safest medicine for the dreadful disease of that cancer particularly. Beneficial in the benign and malignant tumor treatment. Highly useful in curing not only early stage of the cancer, but also every stage of the cancer, it can be the cure. Highly useful in the controlling further spread of the cancer growing in patients. Highly useful for the undergoing chemotherapy, radiotherapy, and hormone therapy. Since it is marketed by Kudus Ayurvedic company, the name has given Kudus CM9. Means the Kudus company, that is Ayurvedic company, has purchased the technology from the CSIR. That means, see, our CSR contribution toward the country, such type of the drug has its given technology given to the Ayurvedic company. That company has already marketed this Ayurvedic drug is at present is available in the market. I given this in the emblem also, that product emblem, where that emblem indicating CSR IACB, this including the CSIR. Not only that one, what CSR, this is I'm talking about my CSR contribution toward the fulfillment of the pharma vision. Or my CSR has taken a patent using the cow urine. They investigated, they studied the more and more research. They found it. Cow urine will be useful for the curing in the treatment of the cancer. Not only cancer, it acts as an antibiotic, anti-infection medicines. Such type of research has been patented by the CSIR. Again, now in that CSIR again, particularly where I'm working in the Institute of Chemical Technology has contributed a lot during the pandemic situation. You know, we the people, ISCT has developed the adjuvant for the covaxin and given to the Bharat Biotechnology. With the support of the ISCT developed adjuvant, the Bharat Biotech able to market the covaxin. See, again, there are two paper statements about the ISCT, their contribution in the pandemic situation. ISCT helped Bharat Biotech developed by the key molecule for the covaxin. ISCT's role in developing covaxin for Bharat Biotech. You know, in that pandemic situation, we cannot order any chemical. We cannot bring them, what are the chemical required from the outside, but whatever available, that is the India's first indigenous COVID-19 vaccine. Means inexpensive and indigenous raw material we used. It's an atom cost and environmentally efficient process. This is ISCT has developed the pandemic situation given to the Bharat Biotech. Then with the support of the ISCT adjuvant technology, Bharat Biotech has introduced the covaxin. This is a major role of the ISCT. But I'm talking about the country contribution. What is the role of the CSR, particularly in the ISCT, toward the fulfillment of the Indian growth, Indian pharma growth in the pandemic situation? This is our contribution. This is ISCT contribution. See, I'm feeling so proud when you're talking about, when you're showing the slide to all the people. My contribution still efficient research is going on to find out the suitable antiviral drug and drug intermediates. That so far I given the Indian pharma vision, then CSR contribution, some of the innovations from independence onwards, then my ISCT contribution. The latter, I am going to talk about the most effective disease in India particularly. That is, one is cancer and the other one is tuberculosis. Every pharma person should know statistics of the present scenario of the cancer and tuberculosis in India. Every pharmacist should know. That is very, very important. Now, let us see the cancer. If you look at a couple of years back, in India particularly, 3 lakhs deaths are occurring in every year. This is a statement given Indian pharma, that means science and technology, based upon the statistics, 3 lakh deaths, couple of years back. But if you look at the present scenario, presently 2,900 deaths per day is occurring. If you look at the total per year, 10,60,000 deaths are occurring just because of the cancer. 3 lakh deaths a couple of years back, but presently, at present, the Indian statistics saying that 2,900 deaths are occurring in a day. Overall, 10,60,000. In spite of the plenty of anti-cancer drugs are available in the market. In spite of that, see, Indian death rate towards the cancer, 10,60,000. How it's a panic? Why it's happening? In spite of the available the plenty of anti-cancer drugs, the only one reason, due to the cost. That means very cost. That means due to the cancer drugs are very cost, expensive, unable to buy. If you look at the Indian population, 80% below poverty, unable to buy. That's the reason. Because of that, keeping the mind, the students, researchers, keeping all these targets in mind and try to contribute adequately for the fulfillment of Indian Pharma Vision 2020. We required, in that statement, there was a last sentence that 20% should be cost effective. Because, see this one. Why? India needs a cost effective drug. Because of, if you look at this one, 10,60,000 drugs are occurring in every year. The in spite of cost effective, that means cost drugs are available in the market. The most of the people, poor people, are unable to buy it. This is the reason. We, the researchers, have to concentrate and frame it, design it, to go for the cost-effective drug. The FDV, the program title itself showing that challenges from academic research to commercialization. Yes, it is possible. It is possible with all our people contribution. For that, just our vision is required. Funding is required. No doubt, government is ready to fund it. When your proposal is up to the mark, no doubt at all. See the Indians, not only by the Indian statistics, this is I'm showing 2,900 a day and 10,60,000 a day, but also World Health Organization, the World Health Organization given a global report. On World Cancer Day, February 4th, World Health Organization has released a global report. That report shows 1.16 million new cancer cases in India. Means, according to their scenario, 7,84,800 cancer deaths are occurring every year. Means 2.26 million 5-year prevalent cases with liquid. Out of 1.3 billion Indian population with breast cancer, 1,62,500 cases. With oral cancer, 1,20,000 cases. With cervical cancer, 90,000 cases. With lung cancer, 68,000 cases. With stomach cancer, 57,000 cases. With colorectal cancer, 57 cases are lying today. Means 10% of Indians to develop cancer, 1 in 15 will die of this disease. How to prevent? So much of science has developed, so much of drugs are developed in the worldwide. Yes, the development is there. Drugs are available in the market, but expensive. Expensive. Where I told you previously also. Again, I'm just repeating that sentence. To look at where the rickshaw pullers and daily wagers, mostly they're getting such type of diseases. Unable to buy expensive anti-cancer drugs. Our aim should be find out cost-effective anti-cancer drug. This would be, if we're able to give that academic research to conversation, the keeping in the mind that that should be cost-effective, where rickshaw puller and deliverers are able to buy it. There, really, we can say our dream, our country dream. That size type of required. The such type of the research is required at present to the Indian farmer. Look at not only Indian statistics, but also World Health Organization released a global report. That global report was showing that so many, 10% of Indians to die the cancer, 1 in 15 is dying. Can't we control? Yes. Let us take the challenge. It can be controlled. Let us produce. Let us develop cost-effective anti-cancer and give it to the country. The first step of the contribution required for the country development, particularly in the Indian pharma vision. Not only that one, if you look at a similar fashion, the tuberculosis. I told you, here only today, I'm just considering the Indian major disease, that the cancer and tuberculosis. Just now we have seen the statistics, not only Indian, but also World Health Organization, so global report also we have seen. Similarly, if you look at the tuberculosis in India, what says? In India, an estimated 370,000 deaths per year. Means 1,000 deaths in a day, 2 deaths every 3 minutes. At present, now it's present. Affects mostly poor people. This is a huge problem to the nation. But how to avoid it? What our Indian missions and science and technology saying that India should be zero growth of the tuberculosis. But how to control? 2 deaths every 3 minutes are occurring, particularly the poor people are suffering with that tuberculosis. Why not? One side we are telling after independence, India has developed 21 drugs. Why not tuberculosis? Yes, tuberculosis drugs are available in the market. Even similarly, like the anti-cancer drug is also expensive, unable to buy. Particularly, if you look at the tuberculosis scenario in India, affects more than the poor people. That more than the poor people are unable to buy that drug. So, this is the reason to avoid this problem. The science and technology has introduced OSDD program, that is open source drug discovery program, that given by the Indian Science and Technology. This is the Indian Science and Technology mission also, to avoid the huge problem of the nation about the tuberculosis I'm talking. What OSDD is doing? That to find out a affordable anti-tuberculosis drug. For that, launched on September 15, 2008. This is a OSDD program. For that, what is the main target? Tuberculosis is the main target. More than 4,500 registered users from 100 countries. They are working for the development of the cost-effective tuberculosis drugs. India has given for the open challenge to the entire world. This is the reason from 100 countries, the more than 4,500 registered and 150 projects and drug discovery program going on at present in India. For that, in that 11th plan period, 46 crores has been sanctioned for the new proposals. So, my dear young researchers, try to design your proposal towards development of the cost-effective anti-tuberculosis drug and drug intermediates. The government is ready to fund you. Are they allocated 46 crore rupees? The fund is ready, but your proposal should be up to the mark. That towards tuberculosis, to find out anti-tuberculosis drug or drug intermediates also. We need not to go for, we don't talk about the final drug only. Up to intermediate also is quite enough. Again, so by involving a large number of researchers so far already is there still to make a difference in the science. This what the Indian Science and Technology Mission, to make a difference in the science. For that, many eminent scientists are playing an effective role to find out the suitable, cost-effective anti-tuberculosis drugs. This is one problem in front of you people to write a proposal, to design a proposal. Get the fund from the then, particularly on March 24th, that is World Tuberculosis Day, when Dr. Soumya Swaminathan was secretary, she told that India is set to launch a new drug to fight the disease of tuberculosis. Why? She told smallpox, measles, or polio had vaccines so far, but there are no such good vaccine for the tuberculosis. For that, the time being, government's next target is to provide one gene expert test kit every district of India. Now it's implementing. You know, it's implementing. See, when she was the secretary of that one, now she told, now she plays a very big world leader now. She's, see the mission. Similarly, I want to give just information. This is AVHS 101, world costliest drug. This is world's most expensive drug, that is, which will be used for the gene therapy drug. This is, so this is United States FDA approved over this drug, approved in the May 24th and 19, used for the one-time treatment of the spinal muscular. But thing is that if you look at its cost, $2.1 million, particularly used in the gene therapy in less than 2 years children. Why I'm giving this information to all my researchers? This is also the big problem in front of you to get the funding. To get the funding. Here I will show this a statement. Okay, $2.1 million, that's expensive. We cannot buy as such. How about affordable pricing in other countries? So this is going to be a big challenge. That challenge has taken. Indian Science and Technology Mission also announced CSR labs with medical institutions should try to develop process and protocols around this gene therapy approach. You sit with the literature, design it, try to submit your proposal to the science and technology, particularly on the gene therapy drug development. You will get the huge funding. Whatever your proposal is, I mean, demand that, even let it be 1 crore, 2 crores. The government is, DST is ready to sanction that amount. If proposal is up to find out gene therapy approach. This is also the big problem in front of our country. Not on our country, in other many countries. Being a CSR scientist, my contribution, I developed so many. I given 12 technologies developed given to the country development. Now, particularly, now looking at your title, challenges from academic research to commercialization. In that title, my work is very suitable for that. So I developed anti-cancer and anti-tuberculosis, some of drug and drug intermediates. In that aspect, first I developed total system of the veratriol and rotodin A, B, C, E, safenafurano, dendrolide. These are the anti-cancer, anti-tuberculosis, cost-effective drugs. So far, I'm talking about they should be drug should be cost-effective. In that aspect, my group has taken the challenge. Then we developed for the total synthesis of that process. Particularly, I have chosen veratriol. Why I have chosen veratriol? There's an anti-cancer, very anti-cancer drug that is. But I given the total how to develop the drug in the cost-effective manner. Why I have chosen instead of so many anti-cancer to develop, the particular I have chosen only particular the first antidote that is veratriol. Why I have chosen means when it's got tested 6-line cell in vitro panel showed more than 100-fold increased potency towards renal cancer with RXF393 cell line, the active towards breast cancer with T47D cell line, with CNS cancer, central nervous system cancer with SNB75 cell line, and prostate cancer with DU144 cell line, with TB leukemia with HL60 cell line, leukemia with CCRFCM cell line, again CNS cancer with SNB19 cell line, and colon cancer with COLO205 cell lines. I thought it, if I develop the such type of the anti-cancer drug, which could be used for the renal cancer, breast cancer, CNS cancer, prostate cancer, tuberous leukemia, leukemia, CNS cancer with a different cell line, and colon cancer. That means one drug is quite enough to cure so many cancers with the different cell lines. That's why showed a more than 100-fold increase in potency towards so many different cell lines. Because of this reason, I selected veratriol to go for the total synthesis. Again, major aim and objective is what is my aim should be cost effective. How we have developed that ones? That means first, the starting material should be cheaper one. We have taken 500 grams of the purchased D-ribose, only 275 rupees, 278 rupees. Now, 2 rupees has increased now due to this pandemic situation. When I purchased, it was only 274 rupees only. Now, this of them 278 rupees now is available in the market. 500 grams I purchased, just 274 rupees. Then I played a little chemistry, gimmicks here. One side, I synthesized veratriol, anti-cancer drug, and their way I also synthesized monotriol. Both are the active isomers. So far, I'm talking drug should be cost effective. No, in that aspect, the simple manner, cost effective manner, we developed this total synthesis. Just the purchase 274 rupees, 500 grams of the ribose. So these are my recent research findings towards cost effective anti-cancer and the anti-tuberculosis resistance. So this is my research laboratory where my students are working on to find out the suitable, cost-effective, affordable, anti-cancer and anti-tubercular drugs. These are my students with the laboratory. With the support, with the help of my eminent, my lovable, my students, where I'm able to produce so much of the chemistry, so many technologies. I show no recent research findings towards the anti-cancer, anti-tuberculosis with their support on these are my research group. This is my great institution where I'm working. There is the Indian Institute of Chemical Technology. A small history about my institution. 1944 is established by the central governments. 1949 found the stone laid by the Pandit Jawaharlal Nehru. Then 1989 renamed as Indian Institute of Chemical Technology. Since we are developing the technology system. This is the reason the name has given Indian Institute of the Chemical Technology. Thank you for your kind attention. Yeah.
Yes.
Yeah, ma'am. If any question is there, I'm ready to just answer it.
Dear participants, if you have any queries, you can interact with sir.
Good morning, everybody. Myself, Dr. Jyotirmayi. The session has given a lot of information, very informative, sir. I really thank you for giving us such good information.
Yeah.
I have a query, sir. For
Yeah,
commercialization, we are up to the mark with academic research, but we are unable to design, we are unable to write a CTD dossier, which is required for commercialization purpose, sir.
Yeah.
Can we get the information about how to give a good CTD dossier for our research formulations, sir?
Certainly, ma'am. For that, what we have to do, ma'am? You please try to select the 1 or 2 good students. Those who are going to stay with you the long time, particularly just nothing but your PhD program. The students should be stay with you more than 5 years. Then the sit with the literature. Nowadays, what industry is looking? Cost effective, again, cost effective. Only thing is you must be able to frame it. Plenty of literature available, but you have to modify. Nowadays, modifications are very much required. That will give, you have to take one just an expert suggestions. Since you are if you are a newcomer, you take some expert suggestions initially, but you should not forget to collect up-to-date literature again. Literature will help you more than 80%. That 20% will help you than an expert. 100% it will be succeeded. What we are doing fundamentally, academic research is nothing but fundamentally we are just at bench level, grams level, mg level. That is the academic research. Once it stabilizes, it optimizes the conditions, we go for the 1 kg batch, 100 kg batch, then we'll go for the commercialization. As per the commercialization is a concern, but the industry has sponsored. Industry should come forward. It is possible. With the collaboration of industry only, commercialization is possible. But at your level, academic basic research is available. Fundamentally, at bench scale, you can develop the technology. For the optimization, for the commercialization, you need 100%, you need support of industry. Then only it is possible. Yes, it is possible, man. Every drug, every research, the fundamentally should be academic research. Where you people are seeing. The particular I visited to Siddhartha. Really, I surprised looking at that laboratories where that people are maintaining. Hyperresearch, being a national scientist. We used to maintain such type of systems, but I could see such international standard laboratories in the Siddhartha Pharmaceutical College, Vijayawada. The recent I visited that college. I appreciated all your staff and your principal and all the faculties. Yes, ma'am. Yeah, ma'am. Thank you. Okay, ma'am.
Sir, I have one doubt.
Yeah, please.
Sir, CSIR has developed Kudos CM9. Very good, sir.
Yes.
So, in which step, what is the possible mechanism? Does it inhibit the angiogenesis or does it inhibit the metastasis like that, sir?
Oh, exactly. That's patented again. Only technology has taken. Already, private company has taken that one. But more than this regarding that drug information, what I have given, no? More than that, beyond that, but I cannot talk. Because that's already under custody of the patented system. And company has taken. That's it only.
Okay, dear participants, any doubts? Okay, sir. Thank you very much, sir. So yeah. Dr. Linga gave a wonderful talk on Indian pharma vision, innovations and their impact. So definitely our pharmacy fraternity, so will get benefits sir, from your talk. So they will motivate and they will initiate and they will start the research work on cancer and also tuberculosis. So definitely we'll get the more funding also from based on this one TB and cancer, sir.
Yes.
So sir has stressed mainly what kind of research is required at present scenario to fulfill the Indian pharma vision. So 14 drugs out of 21 developed by the CSIR. And in first ayurvedic anti-cancer drugs, Kudos QCM5 by CSIR. So this is beneficial in both benign and malignant tumor treatment. So CSIR patented on the molecules from cow urine by the CSIR. And one more effective thing, IACT developed an adjuvant and given to birth antibiotic for the Covaxin. The adjuvant is very important substance for making any vaccine, sir. Innovations required for most effective diseases are cancer and TB. So our aim is to produce the cost-effective drugs for this one. Sir has developed many cost-effective drug molecules for cancer and TB. So thank you very much, sir. Again, we are on behalf of KVSR Scopes and Department of Pharmaceutics and Technology. So we are very thankful to you, sir, once again for readily accepting our invitation to give you a talk on this one as a part of our national webinar. Thank you very much, sir.
Thank you.
Thank you so much, sir.
Thank you. Can I close it?
Yeah.
Yes, sir. Yes. Shall I start the next session, sir?
Yeah. We'll go for the second session.
Good morning, Dr. Murli, sir.
Yeah, good morning. Am I audible?
Yes, sir. Yes, sir.
Yeah, audible, sir.
So hearty welcome to you, sir, for the FDP as a keynote speaker. Dear distinguished and learned participants, I extend a warm welcome to the second session of our faculty development program on emerging trends in pharmaceutical sciences, challenges from academic research to commercialization. I feel honored to introduce our next speaker, a man with potential, industrial experience and success, highly knowledgeable, exceptionally good orator, and a very good human being. A man with a golden heart, Dr. G. Murli Mohan Babu, sir. Advisor Formulation, Dexa Medica Center, Dexa Medica, Jakarta, Indonesia, to enlighten us with his key scientific talk on complex generics, an emerging trend in pharmaceutical industry. Representing the topic, the theme of this topic, emerging trends in pharmaceutical sciences, challenges from academic research to commercialization. So warm welcome, sir.
Yeah, thank you. So shall I start
before
sharing?
that, I'll read out your bio also, sir. I'll start
Yeah.
with the bio. This is just brief introduction of today's keynote speaker. Yes, sir. Just. Let us look into the brief profile of Dr. G.V. Murli Mohan Babu, sir. Oh, shit, just need to start. Share out the sir.
Yeah.
Is it being shared, sir?
Share ayindamma.
Okay, sir.
Yeah, it was shared.
Yeah, shall I start?
Sir is working as a group formulation manager, advisor formulation at Dexa Medica Center, Dexa Medica, Indonesia since June 2015. Dr. Murli's career has taken him through 30 years of study, research, and teaching and administration. Before working with Dexa, Dr. Murli had led the team of scientists at Dr. Reddy's Laboratories, Hyderabad, India, in co-development division of oral solids. Prior to Dr. Reddy's Laboratories, he was working as head formulation R&D of Unichem Laboratories, Goa, India. He also worked with GlaxoSmithKline Laboratories Limited, with Anjana Pharma as assistant manager and formulation scientist, respectively, in the early days of his professional career. Dr. Murli has worked on various types of oral solids, liquids, and semi-solids, controlled release dosage forms, complex generics, herbal formulations, nutraceuticals, liquid lyophilized injectables, oncology products, as well as inhalers for United States, European Union, Canada, Brazil, South Africa, and Asian countries. He has insights in the formulation development of new chemical entities and high-potent drugs, where containment is compulsory during development and manufacturing. He also has rich experience in the field of technology and transfer, manufacturing, science, and technology. Additionally, he has also experience to set up R&D and production facilities during his journey in his pharmaceutical industry. Dr. Murli has completed PhD in pharmaceutical technology and master's degree in pharmacology at Andhra University, Visakhapatnam, India, after completion of his bachelor's degree from the same university. He has published about 8 international publications and 24 national publications. Additionally, he is also credited with 2 international patents and more than 10 Indian patents. I heartily welcome you, sir, as a keynote speaker of this session.
Yeah, thank you. So am I audible? My screen is visible.
Yes, sir. Yes, sir. Your voice
Yeah.
is audible and it's shared.
So my screen is visible, right?
Yes, sir. A little bit, it's down, sir. Please adjust the screen. Yes, sir.
Now it is okay?
Yes, sir. Yes.
Yeah.
Thank you, sir.
Good afternoon, everyone. Thank you for giving the opportunity. I always love to speak to pharma fraternity. And as my origins are from India,
I didn't understand that.
Always I love to talk about my roots and my experiences from education as well as research, teaching, and the people who were surrounded me during my lifetime. So the today topic is a bit complex, because as everyone aware, now the world is moving towards complex diseases. That is why complex generics are required. So what I am going to speak today is what are the what exactly the complex generics and why it is required, and how we are going to formulate the different ways, and subsequently what are the opportunities based on the critical milestones or major steps of development with respect to complex generics to academicians. That is also a couple of slides are there. And also I'm going to boost your spirit by quoting India, what is the benefit and what are the advantages. Now, this is what I am, Murali Babu, now working as a advisor, formulation development at Dexa Development Center, Dexa Laboratories Limited. And these are the details which you can contact me either by email or WhatsApp. I'm always for any suggestions or advices. So today we are going to talk on complex generics. So what we are going to cover before going that, this is a disclaimer. So this is my personal views only, and you should not take it as granted. You should consult relevant regulatory agencies as and when you are moving forward to market or commercialize or even use it. So what we are going to discuss is, we are going to talk about introduction of drug development, and drug development in a overview we are going to talk, because that is the basis for all these generics. In drug development, we are going to talk particularly for the US market, where much clarity is available and everyone is look at US market as a marketplace for the world for developing any pharmaceutical dosage form. And different routes of filing are different routes of product development we are going to talk as part of NDA. And subsequently we are going to talk about generic drug development, what is actually generic drug development, and in that what way contextually complex generics are different. Then we will move to the core subject of complex generics. In complex generics, different types we are going to talk, as well as different opportunities with respect to industry as well as academia, and what is India, how India is gearing up for the world requirements of the pharmaceuticals. So now this is the basic slide, everyone aware what is the drug discovery and drug development process. It starts with discovery, refinement, as well as chemical as well as biological characterization. So that is nothing but preclinical. Then we go with the safety, toxicity in animals, and little formulation development for the animal studies. Then we'll go for the voluntary studies, then regulatory approval once we have a positive results, and marketing, and followed by post-registration monitoring. And based on the post-registration, some lessons will be learned, whether we are going to continue with the product or we are going to withdraw. So in detail, in early drug discovery, what we are going to do, we are identify the target, what target we have to hit, and then we are trying to identify the hit. Then subsequently, whatever chemical or biological moiety we discover, we are going to develop assay method and screening methods. Then during that course, we are going to use throughput screening, high throughput screening, so that number of molecules screened in a shorter time will be faster. Then afterwards, we are going to optimize the lead. Then once this is done, we are going for the preclinical studies. In preclinical studies stage only, we are going to file the IND application. In preclinical stage, we are going to establish the ADME, then some formulation bioavailability factors we are going to optimize. During that stage only, we are going to study the dose range as well as dose titration. Then subsequently, based on these studies, we are going for the clinical development. Then in the clinical development, as you know, the subsequent slide we are going to explain. There are 3 phases: phase 1, phase 2, phase 3, which are very critical, where it starts from the uh, healthy human volunteers, then subsequently it will go for patient volunteers. So number is going to increase. Based on that, number of studies or study centers are going to increase. Usually these phase 2, phase 3 studies are multi-centric studies. And if you look at the statistics, it is about 5,000 molecules. If you start screening in preclinical, it will come out five only from the phase 1, phase 2, phase 3 trials. Finally, one you can submit to FDA. So even after submitting FDA, there will be a chances of failure during post-marketing because of the adverse events. The time period, if you look at almost about 12 years of time period, and the cost now in see, whatever we talk about is new drug application. In new drug, it is completely for the new chemical entities. All studies conducted by the innovator companies, and in that you are going to submit non-clinical, clinical, as well as finally safety efficacy data, as well as quality. Quality is nothing but chemistry, manufacturing, and other controls, including bioavailability, bioequivalence studies if you have multiple doses. Whereas 505(b)(2) is other route where part of the information which is already submitted by the innovator or reference company can be used. However, there will be safety and efficacy studies required, but that is also considered as a NDA. So there are two routes. One is true or pure NDA route. Second one is 505(b)(2) route, where part of the information which is developed for the which is already available in the with the regulatory agents considered while developing a product. This is not completely new product development, means new chemical moiety development, that is in NCE, which is a like dose, coming up with a different dose, or coming up with a different salt, or coming up with a different dosage form, or different dosage form means earlier you have a oral delivery, you are going to come up with a IV delivery. So like that. And also is also called as a NDA, but this route is called as 505. So NDA is like dosage form changes. Like as I said earlier, it is to transdermal or Hello?
Yeah, it is audible, sir.
Yeah, it is disconnected. So 505(b)(2) is nothing but dosage form change or strength change or route of administration change, oral to transdermal or oral to IV. Then dosage regime change twice daily to once daily, or API switch, that is for new salt, a complex racemate or enantiomer or combinations you can use. So overall, if you look at NDA development is very costly. Hence, for the third world or developing countries, whoever it may be, it is not feasible. As I told you, the success rate is 1 in 5,000. Considering that success rate is very low, and development time is high. Now after COVID, maybe like it will be revamped. What is the development time? But however, drug regulators are considering very stringent requirements for the drug development with respect is given by the governments of different countries, various countries, started from US, Europe, India. They started encouraging generic drug development, particularly if you look at US market, where this process of approval of generic drug development started in 1982. So uh, almost 4 decades back, this process is started. As per US FDA, what is the generic pharmaceutical product? So generic pharmaceutical product is which contains same active as in the innovator drug, as well as strength, and also indication form and route of administration should be same. Same are acceptable inactive ingredients. This is also another requirement. This acceptable term is very important because the Q1, Q2 sameness need to be established, and based on the mention or literature evidence, when you are altering the inactive ingredients also, you have to confirm that these inactive ingredients are not affecting clinical uh, uh, any clinical nature of the active. Pharmacologically, these are inert, that need to be confirmed if you are changing the excipients. Now manufactured under same strict standards. This is a container closure system. The label also should be same as the reference product. So usually this abbreviated new drug application is an US generic drug approval for an existing licensed medication or approved drug. So ANDA is the term which is used for the US market. So if you look at major differences between NDA and ANDA, see NDA, as I told you, the submission will be based on the FDC Act 505B, where there are two routes, as I told you, B1 and B2, which is NDA submitted for the new chemical entity or new formulation or combination drugs also for the new indication. So this is the NDA means 505, B1, B2 both are covered. Development approval may take around 10 years, 10 to 12 years. Then see in cost, if you look at it is almost 7,500 crores. It varies from the therapeutic category to category. Then what are the requirements are like you need chemistry, manufacturing, testing, labeling, and inspection reports, like facility inspection reports after you submit the either an NDA, ANDA, which is common, then animal studies, clinical studies, as well as bioavailability studies. Whereas ANDA, the submission is based on the FDC and Act 505J, which is usually submitted for the approved NDA, and also it cannot be filed before 5 years of the NC approval. That is also is very critical time period. Development depends on the therapeutic category, complexity 2 to 6 years. Cost is, yeah, it varies from the 10 crores to 200 crores, which is also based on the therapeutic category. Now, what are the major advantage for the ANDA is everything is same except these clinical studies will be covered by the bioequivalency. So bioequivalency, which is where we are going to establish the therapeutic equivalency or pharmaceutical equivalency indirectly considering pharmacokinetic evaluation, whereas reference product, while they are establishing the safety efficacy, they are going to talk about pharmacodynamic parameters. But whereas in the bioequivalency, we are going to assess the pharmacokinetic parameters like Cmax, AUC, AU. With that, we are going to prove the equivalency. And once we meet the bioequivalency criteria and other requirements as part of the ANDA filing, ANDA submission, then you will be granted ANDA, which you can market as a generic product. So this animal or clinical bioavailability studies are replaced by the bioequivalence studies. This is what is the reason why ANDA is cheaper. Now, coming to the complex generics, before going to the definition and more understanding of complex generics, why complex generics are come into the picture. Basically, complex generics are, as I told you, this started from 1984. Many pharmaceutical companies are focused more on developing simple generic or little complicated generics. But however, these companies are started losing their patent protection or market revenue, then significant, because as the science is evolved and multiple market players are started venturing the generic market across the globe, so competition is increased, and also higher regulatory hurdles as and when there is a new problem come up, usually that will be imposed to the manufacturers. For example, if you know about nitrosamines, which is very recent topic, suddenly the sartans are affected, then followed by metformin is affected. So multiple products are affected because of the new regulation proposed by the regulators of across the globe. So that is also a reason where when you are forced to follow new quality standard, your cost of manufacturing is going to increase. So when there are multiple players, what is going to happen? You cannot dictate the cost. So finally, your cost of the investment versus revenue will be in an imbalanced situation, which is dangerous to the organization. And also filing through 505B1, like NDA route filings are also reduced, like number of approvals are reduced. With that also, there are, if there is new drug, if there is a new drug approval, then only you can able to develop ANDA for that new drug. So if there is no new drug approval, definitely you are not able to file a ANDA. And also another important aspect is like understanding about many generic products is improved across the globe, particularly in India or any other pharmaceutical, major pharmaceutical companies, where the understanding of generic drug development is crossed the limits, crossed the borders. With that, they wanted to venture into the new areas. And also availability of complex analytical techniques. This is also another advantage situation where you can assess anything, anytime within a fraction of time. With that also, so people are industrial scientists are looking at developing a more complex products. So now, what is complex generic drugs? So we talk about uh, simple generic earlier, the pharmaceutical equivalency, as well as physical equivalency, as well as excipient equivalency, finally therapeutic equivalency. This is what. So as per US FDA, a complex generic is a generic that could have a complex active ingredient or complex formulation or complex route of delivery or complex drug device combination. And also there is no clarity in the filing approach. That is also one more additional category. These are all called as a complex generics as per the FDA. Whereas EMA, like European Medicines Agency, they define complex generics as a hybrid medicines whose authorization depends on partly on the results of test on the reference medicine and also partly on the new data from the clinical trials. So here, like you are going to assess reference product in detail, why we have to assess and what is the necessity that we are going to discuss in the subsequent slides. And also the clinical data, what is required for this type of dosage forms are using patient population, whereas for the generic drugs, mostly we go with healthy human volunteers. Though it is bioequivalence study, in complex generic drug assessment, we are going to use mostly we are going to use patient population, which is actually costly affair. So that is what is the, and also the assessment towards reference product is in detail. You have to prove the sameness, particularly with respect to physicochemical character. So those details also we are going to discuss. So however, as it is challenging, time consuming and expensive, so number of players are less. Number of players are less and only few can able to invest on this type of route of drug development, a type of drug development. So as I told you, these are the 5 features. Any drug product can be considered as a complex generic when it contains complex active ingredient or complex route of delivery, or it contains complex formulation or dosage form, and also it is a complex drug device combination, or it is there is uncertainty in the approval pathway. What way you are going to approve also, it is not clear even from the regulatory agencies also. These are the 5 categories where the generic can be considered as a complex generic product. Now we go to one after another. What is complex active pharmaceutical ingredient? So as you know, any dosage form contains API. I think 99% or 99.99% formulations contain API plus excipients. Only I think only one product so far I come across which contains only API, no excipients. So API, what is the purpose of API? As you know, API furnish pharmacological activity, which is the motto of the product which we are taking inside the body. So this API is useful in curing or diagnosing or mitigating or treatment or prevention of the disease. And also this API should not, this is a quoted term, should not affect the structure or function, but it is always based on the risk benefit analysis. Any API or any external chemical agent which you are ingesting, including vitamins, if you are taking more than whatever is body is required, it is going to affect the functioning. But however, that will be considered based on the risk benefit analysis assessed, and if the benefits are more considering the adverse effects, then they are forced to ignore. As you know, earlier cancer drugs, the way the people used to suffer, and currently the way people are benefiting from the formulations, definitely is improvised. So, however, there is an impact of the adverse effects. That is what I mean to tell. Structure is very simple structure, not very complex. Whereas for a complex API, mixture of different components and contain distribution of molecular weight. Also, active components, chemical structure of the API in a repeated nature, means having a range of molecular weight. And also, naturally, there are products containing mixtures containing APIs also considered as a complex APIs. So, coming to the examples, if you come to, so here one last statement is like any formulation which contains complex API, regardless of administration of the dosage form, the drug product can be considered as a complex drug product. So what are the examples for the complex APIs? So mixtures of different components like conjugated estrogens, heparins, botanical drug products such as crofelemer, complex oils and oil derived products. Examples are omega-3 ethyl esters and ethylized oils, which are synthetic polymers like polymer, pentasone polysulfate, savalomar carbonate, inulin, and also metal complex drugs such as ferromoxitol, peptides with molecular weight greater than 40 amino acids having complex higher order structures. Example is liraglutide, calcitonin. Finally, oligonucleotide is also considered as a complex APIs. So any product which contains these APIs definitely considered as a complex generic. This type of API containing products need to be assessed carefully with respect to molecular weight and their physicochemical properties. So that comparison you need to use with respect to reference product, where reference product also you need to assess in detail. So whereas for the simple generics, you may get all this information from the literature, and the techniques used are also very simple. Whereas for this complex generic or complex API containing products, the techniques, particularly analytical techniques used to understand the structure, molecular weight, or its in situ performance, everything is very complex. So assessment is very, very cumbersome. Next aspect is complex route of delivery. So route of delivery, you know. So route of delivery is nothing but way of active ingredient is delivered to the target site of action. So route of delivery is different from the route of administration. That is what you have to understand. Route of delivery may be systemic or local. There are two categories only. So for example, if you consider transdermal products, the route of administration is transdermal, but the route of delivery is systemic. Other way, ophthalmic products, the route of administration is ophthalmic and the route of delivery is local. So this is the difference. So route of delivery and route of administration are two different terminologies which you have to have a clarity. So now, what is complex route of delivery? Complex route of delivery, so route of delivery is systemic. Intended pharmacological action is equally achieved in comparison to the local delivery, then it is non-complex. So for example, you have a pain in the body part, but you take a painkiller like ibuprofen or nimesulide, or that the route of administration is route of delivery is systemic, but however, if any body part has got a swelling or pain, but effect will become a local. So with that, the delivery, the route of delivery can be considered as a non-complex route. But however, when intended pharmacological action can be achieved only with the local delivery of a drug, then that is called as a complex route of delivery. For example, eye preparations, or here it is mentioned as lidocaine patches, which are applied locally to achieve desirable pharmacological action, which is equivalent to the systemic delivery. So hence this is considered as a complex route of delivery. So this is the difference. Like if you can able to achieve systemic, if you can take it orally, but effect can be seen locally, then it is not complex. But if you have to achieve a local effect only by local administration, then it will become a complex route of delivery. If you go to the examples, see all these local administration routes, for example, conjunctival, dental, intracavitary, intracerebral, intraarticular, intracorneal. These are all, the delivery is local, but definitely the complexity considered as a it is considered as a complex route. Whereas buccal, nasal, oral, rectal, topical, and vaginal, these are not considered as a complex route unless otherwise there is a local effect is seen when you are administering the systemic route. This is what is the difference. For example, any buccal, nasal, oral, rectal, topical, or vaginal dosage form, if there is no local effect, and if all effects are related to the systemic in nature, then it is not called as a complex route of delivery. Now, what is dosage form? What is formulation? This is another important aspect which we need to understand. So dosage forms is a physical manifestation, like a tablet which is contains excipient and excipient. Like that, a syrup contains a drug dissolved in sugar syrup and stabilized using antioxidants, or and also to improve the palatability, you add a flavoring or sweeteners. So the physical form or physical manifestation is called as a dosage form. Physical appearance or physical form, or how you are going to administer the product, and also design features affect the frequency. So these are called as dosage form. Whereas formulation is typically it is composition and a process. That is what is the formulation. So typically formulation means it is a composition and process. So how you are going to formulate and what are the components which is called as a formulation. So what is complex dosage form and formulation? Where you are going to administer parenteral drug products, examples, liposomes, long-acting microspheres, or semi-solids and other products are considered as a complex dosage forms. So these are the examples for the complex dosage forms: liposomal formulations, doxorubicin, amphotericin B, vincristine, uh, like that, iron complex formulations also, as well as nano suspensions also, nanotubes also, as well as nano emulsions also, lipid complex drugs also. These are all various dosage forms which are considered as a complex dosage forms. And also one more important point is, oh, yeah, nanoparticle, nanotechnology or nano particles containing formulations never which are taken through oral route cannot be considered as a complex product. This is also the important note. You know about fenofibrate. Fenofibrate, the doses are very high earlier, but after using the nanotechnology or nano sizing of the API, the dose is reduced as low as 45 milligrams. Earlier, it is more around 300 mg. So these type of dosage forms, as generics are means as reference product is available and characterization is much simpler. Considering that these dosage forms still considered as a simple NDA only, not complex NDA. So other examples are suspensions, multivesiculate liposomes, in situ gels, biodegradable microspheres. See, these are all suspension, injectable suspensions are the some of the important categories for though the development is relatively simpler, the development considering biodegradable microspheres or in situ gels, it is relatively simpler. But however, the BE studies are to be conducted using patient population, which will become more complex. When you need to conduct BE studies, whether it is parallel or crossover designs, the issue is like as these drugs are more variable, the number of volunteers and also screening of the volunteer and also getting the patient population which are meeting the prerequisites is very difficult. So that will become a more challenging part in the development. As these are injectable formulations, other important aspect is sterilization. And you have to have a dedicated facility or dedicated equipment, or you have to have a thorough understanding of cleaning procedures, and you have to establish proper cleaning before and after manufacturing the product. These are all the difficult part. And also upscaling is another issue. Whenever you develop a injectable dosage form with where complexity is involved, when you are altering the batch size, definitely the parameters which you need to consider as a critical quality attributes are going to strictly adhere. At the same time, your CMA, CAPPs should also meet the requirements. So overall, what I would like to highlight is the development process is one aspect. Opposed to that, your clinical studies or bioclinical studies are going to be more challenging, then manufacturing is an additional challenge. Hence, the overall number of players in this type of product marketing is less across the globe. So, and also some products which are oil-based solutions, which are, for example, haloperidol oil solution, which is also long-acting, but cannot be considered as a parenteral complex delivery dosage form. Now, other formulation examples are semi-solids, then drug substance may exist in different polymorphic forms, and also creams and lotions. These are also considered as a complex formulations, because semi-solids particularly considered as a complex because the action is local, the effect is local. That is what is the major problem. That is why these products considered as a complex formulations. Now, combination products, drug device combination products. In drug device combination products, you may come across, like for example, simple pre-fill syringes where drug solution is put it in the pre-fill syringe, and whenever you require, you can take it and directly inject it. But these products cannot be considered as a complex. When you have a multiple chambers, when you have specific training required for the administration of the dosage form, so these products are considered as a complex drug device combination products. So device design should have an impact on the drug delivery. That is more critical. So and which is going to have an impact on the drug absorption. So such dosage forms called as a complex drug device combination products. So these are the some example with the dual chamber multi-dose pen injectors, then auto injectors, then oral inhaled or nasal drug products such as metered dose inhalers, dry powder inhalers, specific delivery system with for inhalation route, inotrophic transdermal products, then transdermal and topical delivery systems, metered dose pumps for topical and transdermal formulations, then intrauterine systems, implants with non-biodegradable device parts, electronic device systems, vaginal systems. These are all called as can be part of the complex drug device combination products. Now, this is the last category where the route of filing, a route of approval is unclear. Particularly, these are like abuse deterrent formulations, like where you have a drug abuse, you have to have a antidote formulation. These formulations need clinical endpoint studies. These clinical endpoint studies, you can able to, the data need to be generated based on continuous discussion with the regulatory agencies. So this type of dosage forms also are considered as a complex dosage forms. So the major difference between generic drug product and complex generic is you have to prove Q1, Q2 sameness as well as Q3 sameness. Q1, Q2 sameness is qualitative and quantitative. Usually, qualitative information you can get it from the leaflet of the innovator or regulatory agency's website. Also, you can find out what are the components or what are the different excipients they are using. But you have to establish the quantitative equivalence also. The quantitative equivalence, for example, if you consider polylactide containing microspheres, PLGA containing microspheres, Then PLGA type, molecular weight, its structure, everything you need to prove and establish it is same, whatever the generic company is using as well as reference product is using. So this is what is the major challenge for complex generic drug development. And also the Q3 sameness, physicochemical sameness of the dosage form also you have to establish. So whereas in simple generics, yeah, the advantage is FDA or any EMA also reveal the information of the components which a formulation contains, and also you are allowed to replace to some certain extent where the excipients so-called considered as inert excipient. However, it is not affecting the clinical behavior of the drug. Then you can able to use those different excipients also, but you have to justify. But whereas in complex drug development, you are not allowed, or more or less, it is impossible to use a different composition. You have to continue the same composition, and also you have to prove you are also equivalent with respect to innovator using validated analytical methods, which is very, very challenging. So you have to generate data on reference product, which is very costly, and using a validated analytical methods, proving that reference product which is developed by innovator versus your product, whatever components you have are same. This is what is the criticality. And also this is applicable. However, as it is involves QBD, number of trials are going to be increased, which is also having much cost contribution. QBD principles are is actually statistically driven development, where the number of trials which you are taking are to be properly powered, and also the significance you have to establish, and that too at multiple scales, start from the laboratory scale to pilot to commercial. And particularly for the current validation requirements is more of continuous. So you have to continuously monitor and update the information to the regulatory agencies. So when you are forced to follow such type of development aspects, your cost of product development or commercialization will increase. This is another important in vitro release testing. In vitro release testing, typically for these dosage forms, you have to prove the discriminatory abilities as well as biorelevancy capability. This is also very, very cumbersome process, where your clinical endpoint studies you have to consider to establish batch to batch release criteria for your generic product. So for this, you may force you to validate your or develop your analytical method, particularly for the dissolution, considering multiple factors, numerous trials, because there is no much information available in the literature, unlike simple generics. In simple generics, thanks to academia as well as other regulatory agencies, much information is available. We can use cross-refer and develop the discriminatory dissolution media or any other analytical method easily, even including testing of SA or impurities. These are also sometimes most of the academic institutes only propose methods which are like a starting method for the industry. Whereas for the complex drugs development, complex generic drug development, this information is not available or rarely available, considering that the resource utilization is very heavy. Then other important aspect is pharmacokinetic endpoint studies, particularly in patient population. This is what is the another costly affair. And as it is patient population, patient is screening is very critical. If he's suffering from other disease, usually drug variability is very high in human volunteers. That is why the window of acceptance is given as 90 to 80 to 125 with 90% confidence intervals. Otherwise, the formulator will die with developing a bioequivalent product. Even then, we suffer. To avoid such type of suffering, particularly BCS class 3, class 4 molecules, yeah, regulatory agencies are come up with additional way out, which is like using a replicate or reference replicate or fully replicate bioequivalent studies. With that, yeah, if the variation is because of the API, not because of the formulation, then you can able to justify and relax the limits of the limits, particularly for the Cmax. So, what are the opportunities, particularly for industries? Sorry, particularly for the academia is developing reverse engineering techniques. Though it is costly, maybe you can able to either fund from the government agencies or whoever it may be, but developing a reverse engineering techniques which is going to be helpful to the industry, like understanding of the composition of the reference product, as well as excipient properties, polymer example like polymer composition, structure, molecular weight, and viscosity, or glass transition temperature, and polymer and cap. These are all properties which are going to helpful for the formulator. And also preliminary techniques with respect to formulation and process development. For example, complex products like PLGA based dosage forms, simple techniques of manufacturing, or simple compositions also you can, at least techniques you can propose. Composition maybe you need not to, you should not alter because it is a prerequisite. Techniques you can propose, you can develop. That is another area where you can work. And also science of upscaling and tech transfer, particularly for the complex generics, which is like upscaling and tech transfer for any dosage form is very critical, which is actually, as per my experience, each product has got a different behavior while you are upscaling. This is one more important aspect. Like you cannot consider, like when you are having an experience of tablets of technology transfer of tablets, you cannot consider your experience as granted forever, because each product will behave differently while upscaling. Now I am talking about only about tablets. When you are talking about complex dosage forms, it will become more complex. So considering that, the area of upscaling or tech transfer, particularly for the complex products, is another area which academic institutes can be looked at. Then in vitro release testing, this is another area where you need to have like USP apparatus 3 and 4. You can develop based on the literature. That also will be useful. This is another, it is a supportive studies like pharmacokinetic and dynamic models, which is also useful for formulators to justify their differences or their sameness while developing a complex dosage form. Now, so India. So India now during COVID, like India during COVID across the globe, India established its roots. It is not only during COVID, and before that also, India has got an advantage in the global pharma. That is why we are here. Otherwise, no foreign company hire us. As India got an edge over pharmaceutical technology, and which is always comparable with respect to US and Europe. That is why there is a lot of scope for the Indian scientists, whether it is in India or outside India. So because of this, India is considered as a pharma capital of world. So as everyone aware, India pharmaceutical industry ranks third worldwide for the production volume. Indian pharma was valued about 42 billion USD. That is in 2021. So which is roughly 550 billion USD, around 20-30% we are in cost also. And 20% generic manufacturing is happened from India only with respect to volume. And vaccine supply, we are about 50% across the globe. We have almost 3,000 drug companies, whereas 10,500 manufacturing facilities across the India. And everyone aware about this fact, we have highest FDA approved plants. So why I am highlighting this? Future is yours. Like whenever you look at, or even from the talk of the previous speaker also, definitely the future is future pharma is yours. But fundamentally, one need to understand is the quality of education and also the commitment towards the deliverance. These are the two things which you need to understand. Basically your abilities are going to recruit you into the pass the interview and enter into the industry. But your abilities are your quality of education, whatever you're supposed to get, you should focus and you should learn as much as you can, and definitely you will be entered into the industry or academia. But however, after entering to the industry or academia, your commitment matters a lot. As you know, industry work with a timeline, or industry work with a productivity oriented, and industry never look back at other aspects. That is why we are grown. It is not only the intellectual abilities, but also our pharma generic manufacturing companies has got a very dynamic leaders who never look at watch and always push the subordinates or colleagues to the maximum extent to get the productivity. So for that, definitely one should be ready. So you cannot compare with our industry to any other industry. Definitely there is a starting problem. You will get less paid. That is another issue which you have to be mentally prepared. But however, once you start growing, once you are capable, definitely sky is the limit. Other aspect is morality, discipline. You never try to lie or never try to manipulate any data. This is what one issue has happened around 2010, between 2005 to 2010. Then post to that, FDA and other regulatory agencies are increased their scrutiny with respect to data integrity. Then pharma companies also realized, pharma people means working force also realized the importance of the data integrity, and so everyone geared up. And honesty is the mantra for our pharma. You should go with the data. You should not manipulate the data. Always you have to commit to the black and white only, not to anything. Like in certain cases, maybe your senior is going to force you, but however, you should not take any such heinous action, because it is not only that day, it is going to have a long term impact. And that if any manufacturing facility is suffered, entire cycle is going to suffer. Like one manufacturing facility, then followed by marketing, to backward. If you look at R&D, everyone is going to suffer. Supply chain, analytical, supportive function, analytical, everyone is going to suffer. There are many examples. Start from Dr. Reddy's, Ranbaxy. Before that, actually Ranbaxy, then Sun Pharma. Everyone has got a setback. But however, thanks to industry leaders, all owners of industries are very strong, and they are committed to reshape the pharma industry, and they heavily invested on the instrumentation or upgrading the facilities, and also training the people. Training the people also very important. And also the beauty of Indian pharma community or fraternity is like we are always ready to learn, always ready to learn. So this is the motto. Like you never underestimate yourself, particularly when you are part of pharma fraternity. You never underestimate yourself. There is a lot of future, but however, as I told you, there is a starting trouble. That starting trouble you have to surpass. Now, why India? See, why I'm telling all these things is the India pharmaceutical industry is going to reach 130 billion mark. That is by 2030. So 11 to 12% compound annual growth rate is not small. So it is huge figure. So considering that in another 6 to 7 years or 8 years, there is a growth which you need to be part of that. And this is going to happen because we have good leaders in capital investment. I have seen international companies also, means companies outside India or within India, even small companies also in India, they are ready to invest on the technology. They're ready to train. They're ready to send our people abroad for the training and all. So this is the advantage. So considering that our growth is never be on paper. This is always going to happen. And also we have die-hard leaders. That is another aspect. People struggle and never look back. They meet the expectations as desired and expected. And as I told you, manpower is adaptive, responsive, and responsible. This is another beauty of Indian pharma. Unless otherwise, this manpower is readily available and they are very much adaptive and also very responsive with respect to requirements. In my earlier days of career, we used to work almost 12 to 14 or 16 hours. Only go for sleep, come back and start working. Go for sleep, come back and working. And months together, we have used to be at a manufacturing facility. Ignore about festivals, whatever it is. Only thing is like objective is the filing, manufacturing commitment or filing commitment that we need to meet. That's all. We need to take number of batches at manufacturing facility. Unfortunately, if there's a issue in the upscaling, then you are gone, and you have to solve it, and you have to come up with a process which is reproducible and can be filed. So these are the days where we sacrificed our life. Now we are enjoying. So what I wanted to highlight is you devote yourself in the beginning of your career. Don't look at and don't compare with any other people. Only you devote yourself. Definitely, industry will take care of you. That is another point which you have to keep it in mind. And also across the globe, the cost advantage is there in R&D as well as manufacturing in India. Also, we have technical edge in API and manufacturing process development. Our reverse engineering capabilities are very strong. Particularly chemistry, we are strong with respect to patents. We are strong, and also other allied pharmaceutical sciences also we are very strong. With that, this is the end of my presentation. So this is the last point. Uh, we are also very aggressive in partnering or acquisitions. There are so many partnering and acquisitions are happened. So with that, we are going to achieve our growth rate continuously. So that's all. Any questions? Your voice is not audible.
Dear participants, any any queries? Sir, is it audible, sir?
Yeah, it is audible.
Any queries from participants? Sir, today we had very excellent talk on complex generics and emerging trend in pharmaceutical industry by Dr. G.V. Murali Mohan Babu Garu. So we are very thankful to you, sir. Sir has explained elaboratively the generic drugs and branded drugs with examples and the differences between generic drugs and branded drugs. Sir has explained in detail how NDA and ANDA filing requirements and also the business drivers for complex generics development. So he has explained as for US FDA and European Medicines Agency. Sir also explained the differences between the API and complex API with examples, complex route of delivery with plenty of examples. So the development approaches, dosage form and formulation aspects with examples. Sir has covered the complex drug device combination products with examples. So he also stressed on major milestones of complex generic developments, Q1, Q2 sameness as well as Q3 sameness, quality by design principles, in vitro release testing, bioequivalent studies also. Sir has mentioned the opportunities for academic industries to the industry. Then the India definitely, one thing, the India is the capital for diabetes, as well as the India is the pharma capital of world in respect of generic drug production, as well as the vaccine manufacturing. So definitely, sir, we will follow your advices, sir. So thank you, sir, very much once again for readily accepting our invitation to deliver a talk. Thank you very much, sir.
Yeah, thank you.
Thank you so much, sir.
Yeah, thank you.
I
I will sign off.
honestly appreciate your presentation, sir, and this is going to be stay with our in our memories for so long years to come.
Yeah,
We are pleased to have you, sir.
Hello, Murli Mohan Garu. Good
Hello, madam.
afternoon. I am
Good
Dr. Bharati, your junior at Andhra University.
I know, I remember.
How are you,
Bonara?
sir?
Yeah, I'm fine. How are you?
Okay, sir, I'm fine.
So where are you? Are with Siddhartha only?
From the beginning, my career,
Okay,
total
good.
career in Siddhartha only.
Good. Yeah, so many students you are producing and which are who are doing very well in the industry.
Yes, sir, thank you. Sir, now presentation is very good, sir. Nice
Thank you.
talk.
Sir, there is a query from chatbot, sir,
Yeah,
from
please.
Dr. C. Ramya Garu. She's asking that is complex generics require clinical trials? Please clarify, sir.
Yeah, complex generics, no, not required clinical trials. That is what is the advantage. Extensive physicochemical characterization of the innovator is required. That is one major requirement. and pharmacokinetic endpoint studies in patient population is other aspect, is a requirement. So there is no clinical studies requirement.
Thank you, sir. Thank you so much for your clarification, sir.
When any clinical study is required, it will go to the route of 505(b)(2). And specifically, yeah, when for this complex genetics, you require clinical studies when there is a change, particularly release profiles, or you are altering the composition using any other excipient, or you are not able to prove the sameness, then it require clinical studies.
Thank you, sir. Thank you very much, sir.
Thank you very much, sir, for the tremendous scientific talk we had today.
Yeah, thank you. So shall I sign off or any other questions?
Yeah, you can
Yes,
sign off.
You can sign off, sir. Thank you so
Thank
much.
you.
Thank you.
Okay, dear participants. So today we are closing the session. So tomorrow we will meet again at the same time at 10:30. So tomorrow we will have one eminent speaker, Dr. Girija Patro. He is the head of NDDS department on NAPRO Life Sciences, Maharashtra, Mumbai. He is going to deliver a talk on novel approaches in liposomal formulations and industry perspective. So till then, bye. Have a nice day. Thank you for your registration and your participation. Good luck. Over to Arifa.
Thank you, sir. Thank you. Dear participants, we'll meet tomorrow with the fresh scientific talks. Signing off.